Hi, my name is Amin Zargar, and I'm a postdoc in Jay Kiesling's lab at the University of California, Berkeley. And I want to talk to you about this dynamic regulation system that I've been building in Saccharomyces cerevisiae. So, autonomous gene expression is especially important in um, metabolic engineering because it enables you to go from a growth stage where you're accumulating biomass to a production stage where you're making your target biochemical or protein. And what you really want in this kind of like system, ideally, is something that's easy to use, that's tunable, that allows you to switch from the growth mode to the production mode at virtually any cell density, and you want to do it without the use of costly inducers. So a really amazing system that was set up a few years ago, maybe almost a decade ago, was by Claudia Vickers' lab in Saccharomyces cerevisiae, and this uses the yeast mating response system. So normally the system uses this. The system communicates between A and alpha types, between alpha factors and C2 receptors. And what she did was she went ahead and used this went ahead and expressed the C2 receptor. I'm sorry, she went ahead and expressed the MAT alpha um, uh, the MAT alpha production promoter in one cell, and so it became essentially a self-inducing cell, an autonomous cell, using this yeast mating system. And so what you could showed was that her group showed was that they could turn GFP on at certain cell densities, but this mating factor system relies on the expression of FAR1, and what FAR1 does is it inhibits cell growth as part of the mating factor response, which is not what you want to do in biochemical production. So she didn't show that if you knock that out, the cells will continue to grow, but the problem is that the response of the GFP response is not sustained. It's dropped quite significantly. So the overall fold change was somewhere between 5 to 7 fold at maximum. And so this really just is not enough to produce a lot of biochemicals. Ideally, what you would want is something that's robust, that goes from off to on, that you can use with any promoter is what you ideally want, and you want the time of induction to vary. So either at low growth, low cell density, high cell density, whatever you have in between you. So that's the ideal, right? Any promoter, any cell density from an off to on mode. So what we wanted to do was build off of that work that her group had shown by using different strength promoters on the MF alpha and the ST2. And Wendell Lim's group had actually showed that if you adjust the expression of these alpha factors and receptors, then you could get slightly varying degrees of GFP expression. But instead of just turning on GFP, what we want to do is turn on a recombinase, essentially build a circuit. And what this recombinase would do is it would essentially activate a downstream promoter that would turn on your gene of interest. So essentially, in this example, you have P2 and it goes to a stop codon, so it's not producing anything. But once the system is activated, then the PFUS1 will make the recombinase that will splice out the stop codon and then start expressing gene B. And so the idea is that you would then have that into a metabolic pathway, and then you would have something from not making any product at all to making genes A, B, and C. That would be the dream function and design. That's our goal. So and so by enabling that, you could have things turn on. Like one product could turn on very early. Product one in this example turns on at very low cell density, and product two turns on at higher cell density. So our initial design was we essentially did not put in the production of that gene that makes the alpha factor, and we exogenously added alpha factor because that would make it a lot easier to understand and build our system. And instead of an output plasmid that puts in a biosynthetic pathway, we just had a fluorophore. So essentially it goes from making GFP when the system is not activated to making M-cherry when the system is activated. So that's our system layout. And so our first, we used this PFUS1 promoter that is native. This is how the native system worked. And we added alpha, we essentially had a pre-culture and we added alpha factor and didn't add alpha factor and looked for response. And so what I'm showing you here is, the thing to pay attention to is the input and output. And we're looking for a transition from making GFP to RFP. And what you can see is that the M-cherry production at 0 hour is already making a lot of M-cherry. And so this was supposed to make essentially only making GFP. You can see some GFP production, but a lot more M-cherry production. And this kind of gets amplified at 24 hours. So essentially the basal expression of the native PFUS1 promoter is too high. We wanted to have only GFP expression at 0 hour and then only M-cherry expression at 24 hours. And you can see that that is not what happens. We were already making M-cherry at 0 hours. So we need to get a different PFUS1 promoter. We need to get a different system. So what we did was we looked in the literature and found this PFUS1 J2 promoter. And this was shown to be more robust with higher fidelity than just the native PFUS1 promoter. And we also found these UBIM degradation tags. So this would prevent leaky expression from going on for quite as long a period of time. And using the different combinations, we'd have the original PFUS1 and then with the degradation tag, and this new promoter also with a degradation tag. And so what we're looking for again is something that makes GFP at the beginning and no M-cherry and then switch it. So you can see here that at 24 hours, the PFUS1 J2 UBIM, the new one with the degradation tag, does not make much M-cherry at 0 hours. Everything else does. Everything else has too high leaky expression. And if you go to 24 hours with alpha factor, you can see that it recovers the amount of M-cherry. So the PFUS1 J2 with the UBIM degradation tag has those desired characteristics that we're looking for. So now we wanted to go ahead and characterize this system, and what we want to see is, depending on how much alpha factor we have, what is the response. So with increasing amount of alpha factor, we want to see the transition from GFP to M-cherry, and that's exactly what we do see. With no GFP, no alpha factor added at 24 hours, you essentially get all GFP, and then with 10 micromolar, you essentially get all RFP. And you can see the saturation of the circuit happens at 10 micromolar. And you can see this very clearly in the histogram as well. You can see the switching from GFP to M-cherry with increasing amounts of alpha factor. So now we just need to engineer a control module. So all of this was with us adding alpha factor exogenously. But now what we want to do is we want to add in the expression of that MF alpha factor endogenously. Like the yeast will make it, and then once it gets to a certain point, then the system gets activated. Well, the problem with that is, since it's a irreversible switch, any premature activation during like the cloning phase or something would destroy your circuit. So we're gonna need a control module to prevent that. And so we decided to work. So essentially, the control module would essentially be that when we're going to use this bar1 protease, and so essentially before we decide to activate our system, before we're ready to actually make stuff, we want the bar1 to be made to degrading any alpha factor, any premature activation, and then afterwards we want the bar1 to not be made. And so what we're going to use is doxycycline. And so essentially we're using an induction system to control production during the cloning phase. And then during the production phase, we don't use this. So only at very small volumes do we actually need this doxycycline. So first we test, so essentially just to reiterate, in the cloning phase, doxycycline will activate bar1 and that'll silence the circuit. In the production phase, we won't add doxycycline and that will allow circuit activation. So, first we just checked whether or not the doxycycline system will work. And so you get blue fluorescent protein if you add doxycycline. It does work. this is in the literature, not surprising. But now we felt comfortable to going ahead and seeing if we could get the system to work in our system. So, so this is the setup. We essentially had overnight cultures, and we induced them, one of them with bar1, the wild type, with and without doxycycline, and we add alpha factor to these cultures. So the bar1 that's induced by doxycycline should degrade that alpha factor, and when it doesn't have doxycycline, that alpha factor should stick around. And then instead of, normally we add alpha factor to our circuit, in this example, we're just going to add this supernatant. So the bar1, if it was induced with doxycycline, when it gets exposed to the alpha factor, it'll degrade it. If you add it to the circuit, it shouldn't become activated because the bar1's degraded off the alpha factor. If you don't add the doxycycline, then the alpha factor is not degraded. So then if you add that supernatant to your strain with the circuit, then it will be activated. And so that's what we're looking for, and that's exactly what we see. So what I'm showing you here in the gray is the normal way the circuit works with exogenous alpha factor. You can see that without alpha factor, you just have GFP expression, and then with alpha factor, you get this expression of M cherry. And then you can see that this holds true with our setup of essentially adding the supernatant that is induced by doxycycline, and alpha factor. So you can see, that the dynamics between the original circuit and the circuit using doxycycline to control bar1 mediated control of the alpha factor also works. I know that that's kind of a mouthful, but essentially you can just see the dynamics are the same here, which is what we were looking for, that our control module works regardless. So that's very exciting. So now we can go ahead and actually put in the full system. So here we have the MF alpha factor being made by a variety of promoters, and the ST2 being also made by a variety of promoters, 4 promoters each, and you have this plasmid. Now it would be controlled by doxycycline during the cloning phase, but during the production phase, you would not add doxycycline, the system would be activated. So this will be 48 different combinations because we're using the 4 different genomically integrated promoters of MF alpha, the 4 different genomically integrated promoters of ST2, and 3 different systems of response. So essentially we added another feedback loop for production of MF alpha if we just weren't quite getting enough MF alpha. And so we've done some testing on that, and what we found is that at 24 hours, all cultures were flipped from GFP to RFP. We're optimizing this right now to get this to be exactly the way that we want it to work, and we're very close to getting that. And so so our goal is to show the tunability of this system with different fluorophores, where we want to show that the different levels of MF alpha and ST2 can result in different, at different time points for the expression of the system. And then we're going to translate this to obviously the production of actual biochemicals. We think this can be especially valuable for biochemicals where it has toxic intermediates. And our goal is to establish this mathematical model that actually really helps characterize our circuit. With that, I just want to thank Jay Kliesling, who's a fantastic advisor, Leonard Katz, and Clem Fortman, who are just great scientific people to come to for mentorship and help. Jenny Landberg, who was a visiting PhD student who did so much of this work with me, and just an absolute joy and pleasure to work with, and my wonderful undergrads, and of course, the Keesing Lab. Thank you, and I really appreciate your time.